it was fibrinoid radiation or necrosis. There was no tumour there, in fact, despite the appearances in imaging. And this is, of course, pseudo-progression, which happens in a significant minority of these cancers. So having new imaging tests to be able to discriminate between what is true tumour progression and what is pseudo-progression is absolutely key. Another absolutely important element for why we need imaging is this: heterogeneity. It's a feature of all tumours, shown quite nicely on the right-hand side by that classical New England Journal paper showing the heterogeneity within renal cancer. And it exists on multiple levels. It exists within a tumour, it exists between tumours or between metastases, between patients. It exists in space and time, it can be structural, functional, and metabolic. And that's part of what this talk today will be about. And imaging is absolutely key for detecting heterogeneity, whereas some of it may be possible with circulating biomarkers, and we get unique insights with imaging. And just as an example of that, before I get on to metabolism, this is a study that we completed a little over a year ago in metastatic melanoma with our colleagues here in Cambridge, where the patients underwent immune checkpoint inhibitors, and we did multiparametric MR baseline before treatment at 3 weeks and 12 to 14 weeks after commencing that treatment, looking at how that structure and function may change over time. And this is the work of Doreen Lau, who's now in Oxford. And as an example of this heterogeneity, this is a single patient, 4 lesions over time, so baseline 3 weeks and 12 weeks. Patient obviously had one form of treatment in that time frame, but one of the lesions progressively got smaller, one got bigger and then smaller, so that's pseudoprogression that I talked about. This got even bigger and even smaller, and then a 4th lesion, which largely stayed the same. So we were trying to find ways of teasing apart these responders and non-responders, the pseudoprogressors. So we looked at diffusion initially, and this DAP, or equivalent of an ADC. Even at 3 weeks, we were able to discriminate between the pseudoprogressors and the true progressing lesions. In fact, we even see some changes in the T2 weighted imaging in the pseudoprogressors at that early time frame. So the diffusion changes, the changes in cellularity are happening relatively early. Later down the line, so 12 weeks, we started to see changes in DCE MRI and perfusion and membrane permeability, where we again can start to discriminate between the non-responders and the responders. So this pattern of where we get early cell changes, later changes in the microenvironment around the tumor, ultimately changes in size, are really the characteristic pattern and the way that tumors change over time. Can we go earlier? Are there things that we can look at even before their cell changes? And I guess that's what a lot of this talk will be about. But before I get to what those things may be, metabolism being one of them, just one final way where we can look at heterogeneity and how it can link with other biomarkers, which I referred to. So this is a bit of work that we did a few years ago comparing ctDNA, again in metastatic melanoma, and how that related to the heterogeneity on imaging, this time in CT, and showing a correlation between the two. So taking circulating biomarkers, linking them with imaging biomarkers, we can better phenotype our patients, better understand who will respond and who won't respond to therapy. So I'll now turn to metabolism, which, as I said, could this be an even earlier metric of response to therapy? And it is, of course, one of the canonical hallmarks of cancer, the deregulation of cellular energetics. And metabolism changes in a number of different ways in tumours compared to normal tissue. Classic, of course, being aerobic glycolysis, the Warburg effect that I'm sure all of you are familiar with. But there are other ways in which metabolism can change. For example, alterations in lipids, alterations in amino acids also occur in many tumours. We know that tumours are almost always acidic. That's partly because they produce lactic acid, partly because there are changes in the buffering status, that we have altered perfusion into the tumours. We know that a number of tumour suppressors and oncogenes control metabolism, and there are some rare examples of where metabolism can be modified as a treatment, not many, but some. And then finally, there are a few very, very rare examples of where some mitochondrial enzymes are tumorigenic, and I'd like to talk a little bit about those now, particularly isocitrate dehydrogenase, fumarate hydratase, and succinate dehydrogenase. So the first, isocitrate dehydrogenase, produces an oncometabolite 2-hydroxyglutarate. So this is a metabolite which can engender tumours. It's produced at relatively high concentrations. And this is a beautiful example from the UT Southwestern group showing how that can be picked up with spectroscopy. And this is one of the real powers of MR, the fact that we can use resonant frequency of a nucleus within a molecule to show the fingerprint of it, to be able to detect it non-invasively. I mean, that's really one of the great strengths of MRI. So we took this concept and looked with some of our endocrinology colleagues, and this is the work of Ruth Casey, looking at succinate dehydrogenase mutated tumours that produce GISTs, paragangliomas. This is a paraganglioma here that you can see on this image. And here we were able to show succinate levels at 2.4 ppm. In fact, we even were able to use it where the clinicians thought there probably was an SDH mutation, but they couldn't demonstrate it using conventional sequencing. And we were able to show a functional mutation there because there was succinate produced here that you can see. And they went on to show that this was due to hypermethylation of a promoter region of SDHC. So the functional imaging led to the discovery that there was, in fact, a mutation within the enzyme. And then finally, can we use it for treatment response? And here we have an example of pre and post sunitinib treatment in a metastatic GIST patient. Unfortunately, this patient didn't respond to therapy, and we see an elevation in that succinate, but it shows how that specific molecule can be used to monitor therapy. And then a final example, this is something we did more recently, looking at fumarate hydratase in Reed syndrome, which is hereditary leiomyomatosis and renal cell carcinoma. On the left-hand side, we have an example of a young female who had multiple skin lesions, which were thought to be leiomyomatosis. She had a uterine fibroid, as you can see here in this image, and we did spectroscopy. And at 6.54 ppm, we were able to demonstrate the presence of the fumarate here. And ultimately, that was shown to be a missense variant in exon 8 of the fumarate hydratase gene. And just a few weeks after that, very similar patient turned up to the endocrinologist saying she was 45, she had multiple skin lesions, she had a history of a hysterectomy, so we don't know whether she had a fibroid, and a large mass was found in the retroperitoneal region, which was felt, could this also be a tumour with elevated fumarate? Now, we were quite disappointed at the time that we couldn't find any fumarate. We thought maybe it wasn't sensitive enough, but in fact, this patient transpired to have a non-malignant condition, a hyaline vascular Castleman's disease, in fact, didn't have the mutant FH gene, showing that using our functional metrics, we could distinguish between the FH mutation and something that was not. Now, those are pretty rare tumours that I've just cited in the last three examples. Ideally, we'd like to find something which is more widely applicable across the board, which could be used in the majority of tumours. And of course, aerobic glycolysis is an example of aberrant metabolism that occurs in a large number, if not all, tumours to some extent. And therefore, can we harness this reaction of pyruvate to lactate under the action of lactate dehydrogenase with the concomitant exchange of NADH to NAD? So, this is a really key reaction that I'm going to talk about in much of the next bit of the talk. Now, those three molecules that I mentioned before, the 2-hydroxyglutarate, fumarate, succinate, are present when there's a mutation at very high concentrations. They're in the millimolar range. So they're relatively easy to detect with conventional proton spectroscopy. The vast majority of our other metabolites are at much lower concentration. And so although the great strength of MR is non-invasively being able to detect these metabolites, its great weakness is its insensitivity. And so if we take an NMR-active nucleus within a magnetic field, in essence, we generate signal from about 5 parts per million of the available nuclei. So we're discarding information from, let's say, 999,995 of the nuclei. So if there was some way of trying to harness those other nuclei, then we may be able to increase our sensitivity for detection. And hyperpolarization is a very generic way of doing that by forcing the nuclei into one or other energy state and therefore increasing the sensitivity. Now, there are many ways of hyperpolarizing, and one of them is dynamic nuclear polarization, and that's the technique that we've been using in Cambridge now for more than 10 years. And we take carbon-13 labeled nuclei, in particular carbon-13 labeled pyruvate. We dope it with a free radical. We cool it to 1 Kelvin or minus 272 degrees C in a magnetic field, usually 5 Tesla for the clinical device that I'll talk about in a second, and it's irradiated with microwaves. And this is the preclinical device in Kevin Brindle's lab that we originally used as part of these studies, and I'll show you some data from that in a second. And then the clinical device on the right-hand side, so it's called a spin lab. So the hyperpolarized pyruvate is then warmed very rapidly with warm pressurized water, and is then injected into cells, into animals, and ultimately into humans. And it's the real-time formation of that lactate that is the powerful tool in all of this. So this is the path that we've taken towards clinical utility in Cambridge. And I'm just going to talk about some of these areas and how they've been important. And then I'm going to move on to show you some of the clinical results. So first of all, the pharmacy. I mean, that took some time to set up. And a big shout out to our team here in Cambridge who have developed this. It's a specialist laminar flow hood. We use a liquid nitrogen, gaseous helium, a laser welder. It's all done within a clean room environment. And this has taken some time to develop in terms of how we can get reproducibility of our pharmacy kits, such that in the last 3 years or so, we have got approximately 90% success rate with these kits. So it's really been a huge amount of work to do this, but the success of it has allowed us to take this into clinical studies. We've obviously been hit by many things, including COVID, over the last few years, which have delayed some of these studies. What about acquisition and hardware? I think it's absolutely key the way that we acquire the data. On the left-hand side, you can see how we've done some of our breast studies, and patient comfort is an absolute priority. On the right-hand side, some of our coil systems that we've been working on in conjunction with Rapid Biomedical and others to try and produce images which are homogeneous across the abdomen and pelvis, to produce images which are not dissimilar to what we can do with PET. You can see a sodium MRI image of the abdomen and pelvis there, and that's taken with a sodium equivalent coil that we're now trying to transition across to C13 in conjunction with Rapid. So these sort of coil systems will be key to be able to produce quantitative metrics in order to be able to measure changes over time and discriminate aggressive or less aggressive disease. And I think it's really key that we as a community should try and harmonize our protocol acquisitions. Analysis is obviously key. Just 2 examples here. On the left-hand side, this is some work we did with one of our PhD students a few years ago, Charlie Daniels, looking at segmenting tumours based upon metabolism. So taking areas of high metabolism in an unsupervised way and separating them out from the rest of the tumour. And potentially this could be a powerful tool for delineating areas of tumour that I will talk about later on. On the right-hand side, more recent work, and this is in conjunction with our maths colleagues, Matthias Erhardt, looking at super resolution, where we take a relatively low resolution metabolic image. And that's one of the challenges that we face with this technology, that's relatively low compared to some of our anatomical imaging. Combine it with the anatomical imaging, use that to enhance the output using, for example, here, structure guidance in order to produce increased metabolic resolution. Potentially that would allow us to delineate more clearly areas of high and low metabolism within the tumor. Validation is key, and that's technical validation, repeatability, reproducibility, but also biological validation. And that's something that we in Cambridge are very keen on trying to explore further. In this example, we're doing that in a kidney tumor where, with the work of some of our colleagues, such as Maria Crispin-Ortuzza, we've got a mold of the tumor, and the tumor is taken out in surgery, it's put into the mold, it's sliced, as you can see here, so that we can correlate very directly the pathology that we're getting with the imaging that we've acquired pre-surgery. And so we can then take multi-regional samples and understand how, on an individual voxel level or a group of voxels, how that correlates to the biology within tissue. And then taking a whole sort of set of tools, we can start to probe the tissue from H&E through to immunohistochemistry. These are spatial transcriptomic images of the enzymes. So studying transporters and enzymes that are really important in pyruvate metabolism. And then examples of other tools are shown on the right-hand side. We're also looking at mass spec imaging, proteomics, and other tools to tease apart the metabolic fingerprint of the tissue. And then finally, what are the applications for these? How do we compare to other techniques? And really, that's where much of the rest of my talk will center upon. So that's the key reaction, pyruvate to lactate. What can we do with it? Well, in the field of oncology, there are two major areas where I think this will have a role. The first on the left-hand side is looking at treatment response. So this is some of the very early work from Kevin Brindle's lab looking at treatment response in lymphoma. And on the left, we have the baseline lymphoma tumor, very yellow, lots of metabolic activity there, lots of lactate. And after only 24 hours of etoposide, we're seeing a very significant reduction in that lactate labeling. So really a metabolic shift happening within hours. And that goes back to my comment about the metastatic melanoma and can we look even earlier than that 3-week time point. And then the right-hand side is the other major area where I think this will have a role, and that's to grade tumors or to look at aggressiveness. And this is work from UCSF, John Kanovich and colleagues, looking at how you go from normal prostate through low-grade and high-grade tumors, and we get an increase in lactate. So can we non-invasively stratify these tumors based upon their metabolic phenotype? So some examples of how we've done this in Cambridge, and this is a prostate example. So now in humans, compared to what you just saw in the animal model, and this is work by Nikita Shushintsev and my colleague Tristan Barrett in radiology, where we're looking at here how you go from clinically less aggressive to clinically more aggressive prostate cancer, looking at the percentage of Gleason pattern 4 disease as a metric of that, a clinically significant metric, where as we go to more aggressive disease, there's a switch to glycolysis. And that's seen down here in the bottom left, where you correlate the Gleason pattern 4 disease with the amount of lactate. So the lactate can tease apart the more aggressive tumors. Now, what does that mean in terms of biology? Well, Nikita was looking at the enzyme involved, the LDH, using spatial transcriptomics, and we get more LDH in the higher grade tumors. But also we see some metabolic compartmentalization. So the MCT4 is what exports the lactate. And as we go from low to high grade tumors, we get a shift from stroma to epithelium of the expression of that transporter. Indeed, we also see a switch to from the LDH and the PDH. The PDH is what's controlling the entry into the TCA cycle. And by using the LDH to PDH ratio, we can look at the difference between non-oxidative and oxidative metabolism and how that changes going from low grade to high grade tumors. So again, we can use these tools to start to understand metabolism in new ways. Similarly with breast cancer, and this is the work of Ramona Wojtek, where we have 7 breast tumors here. The 5 on the left are grade 3 or more aggressive breast cancers, the majority of which are triple negative breast cancers. And one of them was estrogen and progesterone receptor positive. And they had higher lactate compared to the grade 2. So we're seeing exactly the same thing here in the breast. More aggressive tumors have higher lactate. And what's the reason for that? Well, using immunohistochemistry and RNA expression data, we've also shown that that relates to hypoxia. It relates to the pyruvate transporter, MCT1, that brings the pyruvate into the cell. To some extent, it correlates with size, but it's more than size. And that's probably down to hypoxia driving glycolysis, which increase the pyruvate transport. Now, Ramona then went on to look at how we could exploit this, because all of those tumors which have high lactate are also tumors which are more likely to have neoadjuvant chemotherapy. So 17 to 40% of operable early breast cancers undergo this neoadjuvant chemotherapy, and particularly the triple negative breast cancers. And the advantage of this is that we can downstage the tumor. There's breast conservation, which is more likely, which is clearly important from an aesthetic point of view. It also gives us a window in which we can test novel drugs. And that's really key to us as imagers. So we can use this to understand what's happening for new therapies and potentially moving the patient from a less effective therapy to a more effective one. And also with neoadjuvant chemotherapy, we have the ability to look at pathological complete response many months down the line. We know there's a very good correlation between pathological complete response and good prognosis. So can we use the imaging up front after a very short period of time to understand what's going to happen many months down the line in surgery, and indeed ultimately what the patient's prognosis will be. And so we've been working with our colleagues here in Cambridge, oncologists working in breast cancer, to study this. And our this is a case report initially, which Ramona looked at after one cycle of chemotherapy, 3 weeks of conventional chemotherapy. And in that relatively short time span, we had a reduction in metabolism. But interestingly, the K-trans went up on DC-MRI. Now, traditionally, that's associated with non-response, whereas we would have predicted with the decrease in metabolism that maybe the tumour was responding. And indeed, the volume ultimately got smaller, the patient showed response at surgery. And so potentially could our hyperpolarizer, could the metabolism be revealing something that is more difficult with conventional imaging? So that was just one case, as I said. We then went on to look at a cohort of cases, and Ramona chose to look at a very early time point, 7 to 11 days, so essentially 1 week after starting this neoadjuvant chemotherapy. We have two examples here. One is a HER2-positive tumour, which we saw a decrease in the lactate overpyruvate over time, and then a triple-negative breast cancer where we saw an increase in that rapid time span. So overall, this was the response from the 7 tumours. We had a pathological complete response in those that had an increase in the lactate to pyruvate ratio of 20% or more. Now, volume didn't predict response, nor did the vascular permeability, the K-trans, whereas the metabolism did. Now, this is interesting because it relates to something very clinically meaningful, but the majority of the preclinical studies have shown a decrease in lactate over pyruvate or lactate labeling following treatment. There have been a small number of exceptions to this, but largely it's shown the opposite. So why is that? Why is the clinical study revealing something which may be different from animal studies? First of all, it shows us the importance of doing these early clinical studies. And we know that it's not down to pyruvate delivery because we did the DCE in conjunction with it. And one of our hypotheses is that it was driven by hypoxia or indeed immune infiltration. So our next step now is to try and understand from a pathological point of view what was going on pre and post treatment. So we know that both the MCT1 and LDH correlate with the lactate labeling. And so that's the import of the pyruvate, the exchange with LDH. And at least in our first cohort where we looked at the relationship between expression and individual tumours, we then wanted to explore this in a much larger group. So we took the Metabolic data set, which stands for Molecular Taxonomy of Breast Cancer International Consortium. It's a large genomic and transcriptomic data set. It's about 2,000 breast tumours. So we wanted to try and understand what we could glean from that. And we showed a correlation between, on one hand, the LDHA MCT1 expression, on the other hand, measures of hypoxia, so carbonic anhydrase 9, HIF1 alpha. And actually, this may be more interesting in a way in that these potential metabolic biomarkers, ones that we can measure with the hyperpolarizer, correlated to relapse and survival, particularly LDHA, the enzyme, CA9, the measure of hypoxia on the surface of the cell, were associated with reduced relapse-free and overall survival when overexpressed. So could we, using the hyperpolarizer, measure some important biological metrics which ultimately have some clinical significance? And I guess that's one of the really key things that arose out of this work. So now I'd just like to take a few more examples, a few more exemplars, if you will, of what we've done with this. This is in kidney cancer. It's the work of Stefan Ersprung, who looked at a range of clear cell renal cell carcinomas, as well as some other tumors like an oncocytoma, for example, and showed that the relationship between grade and the KPL as a metric of increased metabolism was that the more metabolism, the higher the grade, just as we've seen with the prostate and just as we've seen in breast. In the case of the kidney, it was related to MCT1. And again, looking at that, there was a relationship between MCT1 expression and overall survival and disease-free survival. So just in the same way that LDH may be related to the overall survival within the breast population, here we're seeing that with MCT1. And I think a key thing for us is now to try and integrate these novel imaging tools into our clinical trials. So this is an example of something we're doing with our oncologists working in renal cancer and surgeons, where prior to the nephrectomy, the patient has experimental therapy for about 14 days, a window of opportunity trial, where they have initially imaging and biopsy at baseline. They have these 14 days of treatment, and then they undergo pre-nephrectomy imaging. And as a consequence of nephrectomy, we obviously get a lot of tissue that we can correlate with the imaging. So can we explore what's happening during that time frame? I mean, another really important aspect of what we can do with our imaging is guiding biopsies. And this is again work of Nikita Shushenstev, who's been looking at whether you can guide biopsies using metabolism. I mean, obviously we can tell the tumor from pathology, but how do we do that in vivo? We can use, in this case, the T2 weighted imaging to delineate the prostate tumor. Could we use the metabolism? So by correlating the metabolic fingerprint, the metabolic shape of the tumor, and how that relates to expression on immunohistochemistry and spatial transcriptomics, we can see how important potentially targeting biopsies to areas of high metabolism could be. And so that would allow us an opportunity where we can overcome some of the sampling errors from biopsy by using the metabolism to guide to the areas where it may be most insightful to gain that bit of tissue. And what about the brain? This is some of the work of Ferdia Gallagher, looking at a wide range of brain tumors or gliomas here. And what was interesting here for us is that we were expecting to see quite correlations between the lactate and the pyruvate and what we're seeing between tumor and normal appearing brain parenchyma. But actually there was a huge amount of heterogeneity between these tumors. Some of the tumors showed lower metabolism of lactate compared to normal appearing brain parenchyma. Some showed higher. On average, in fact, it was approximately the same between the tumor and the normal appearing brain parenchyma around it. In fact, the thing that was very significantly different was the difference in the amount of bicarbonate produced, and that there's much less bicarbonate within the tumor as a measure of oxidative metabolism compared to the surrounding normal appearing brain parenchyma. These patients went on to surgery, they had multi-regional biopsies, and Ferdia went on to show that the LDHA, the enzyme, correlated with this lactate expression. So what this exemplifies is the variation in the underlying molecular drivers between tumor types. In some cases, it's the enzyme, in some cases, it's the transporter for pyruvate, in others, it may be the exporter for lactate, or indeed the expression between cells, whether it's in the epithelium or the stromal tissue, and how that changes with and without treatment and as a tumor becomes more aggressive. So understanding the molecular environment in which this metabolism is generated is going to be absolutely key. Another very interesting finding from this study was that we saw increased glycolytic and oxidative metabolism outside of the tumor compared to volunteers. So potentially the tumor is not only affecting the metabolism within the boundaries that we can see on the proton MR, but also more distant metabolism as well, which is fascinating and we've yet to understand more fully. So we can have a range of experimental metabolic tracers that we could use for these sort of experiments. I've talked a lot about pyruvate. There are other new molecules coming on board which can be used for hyperpolarization. And we can use a wider range of metabolites for giving in theatre, for example, glucose. If we take the organ out and perfuse it ex vivo, even more metabolites can be used in that scenario. And if we're using cells, a very wide range of metabolites and tracers can be used. But there are just a few that we can use in vivo, as I said. And I'd like to concentrate in the last few minutes on one of those, which is using deuterium to label glucose, a technique called deuterium metabolic imaging. And on the left-hand side, you can see the metabolic pathways that each of the two molecules may follow. Glucose on one side, labeled in the 6,6 position with heavy hydrogen or deuterium, and then pyruvate labeled with carbon-13, which I've been talking about over much of this talk. They clearly have different transporters. One is GLUT1, the other is MCT1, into the cell. Once in the cell, they converge on pyruvate, both form lactate, but labeled in different ways. And then they diverge again when entry into the mitochondrion, where PDH on the inner leaflet of the mitochondrion can determine whether it enters the TCA cycle, acetyl-CoA, or in the case of the C13 label, that comes off as carbon dioxide. In the case of the deuterium label, it continues around the TCA cycle to label alpha-ketoglutarate and ultimately glutamate and glutamine. And it's that glutamate and glutamine together, GLX, that we're able to image using spectroscopy. And on the right-hand side, this work that's really been led by the Yale group with their paper in Science Advances in 2018, showing how you could do this both in the healthy tissue and tumor at high field. So these were 4.7 and above. So we've been working to try and understand whether we could take this into clinical field strength, and whether we could do this at 3 Tesla, for example. So this is the first example of doing this work at 3T, led by Josh Kagi and Ali Khan. And here we have examples following oral administration of this deuterated glucose. So importantly, this is much easier to give than intravenous pyruvate. We wait about an hour or so before acquiring these spectra. And the middle example is the spectra that's been acquired from the whole brain on the right-hand side, a slice-specific spectrum. And we can see not only that lactate that I talked about that's labeled with deuterium, but also the GLX. So we can see oxidative and non-oxidative metabolism simultaneously with that. So we thought we'd compare it to what we see with the hyperpolarizer. We thought it'd be quite interesting to try and tease apart metabolism using these two novel techniques. And the example, the results are shown here, because essentially we measure the same thing. We have a lactate GLX ratio with the DMI, and I'd like you to concentrate on what's being ringed here in red. And in the bottom, we have a lactate to bicarbonate ratio, the equivalent ratio of non-oxidative to oxidative metabolism. And the difference between these ratios is really quite stark, 20-fold difference. Now, why is that? Well, some of the results are due to the way we deliver the probe, whether it's intravenous for pyruvate or oral for glucose. Some of it is related to concentration, in that we give supraphysiological concentrations of pyruvate, whereas ultimately the glucose is largely at steady state or close to steady state. The timing is different, as I mentioned, in that the pyruvate is imaged over seconds to minutes, whereas the glucose, the oral glucose, is over an hour or so. Now, the transport metabolism is clearly different, as I talked about, but it's probably down to these 3 factors here, which describe the difference in the way that we can probe metabolism. So together, they potentially have the ability to phenotype not only normal tissue in this case, but also tumours in new ways that we can understand with oxidative and non-oxidative metabolism. So I think with that, I will draw the talk to a close. I mean, I hope I've shown you the potential clinical roles for novel metabolic imaging in oncology, particularly for assessing metabolic heterogeneity. It's quite clear that we're seeing that to vary not only within tumours, but between tumours and patients. We can stratify patients based on metabolism, particularly based around the level of lactate labeling. More aggressive tumours produce more lactate. Can we use that lactate to target biopsies so that we can get the tissue of most interest to the pathologist and link that to what we're seeing on the imaging? I think that will be absolutely key. I've talked a lot about early treatment response. How early is early? You know, what is it that we're measuring? Is that hypoxia? Is that immune infiltration? Is it a true change in the tumor metabolism? Is it within the epithelium, or is it in the stroma? These are uncertain in terms of the questions that we need to answer now going forward with these technologies. I haven't really talked about guiding radiotherapy, but clearly we could use metabolism to guide radiotherapy, given the importance of hypoxia and the relationship of hypoxia with lactate, that understanding that may be key for guiding radiotherapy. If we're using this for longitudinal imaging, because it doesn't involve ionizing radiation, that might be absolutely key. If we want to image patients at risk of radiation, for example, children, women of reproductive age. And I started off by talking about the circulating biomarkers. I think a lot of what we're doing should be complemented not only with circulating biomarkers, but also with pathology. And so linking imaging, pathology, and circulating biomarkers really is the way that we should be moving forward to more fully phenotype patients. And so with that, I'll say thank you to our big team of people who've made all of this possible. This is just some of them here at one of our recent celebrations. So thanks to our team, and I've talked about the people who have undertaken the work as we've gone along. And thank you, of course, to our sponsors and our funders. We're very grateful for all the money that they've given us to make this possible. And thank you for listening. Very happy to answer any questions that you might have.